Hi there. I'm really glad that you're following this tutorial on building bioinformatics workflows using Snakemake. In this video, I want to show you how you can effectively use the resources that we will be using throughout the series of tutorials. Now, one of the most fundamental resources that you will access, and I will include the link in the video description, is our GitHub page. Now, when you head over to our GitHub page, you will find this project repository called the Schwan project. Within this repository, you will find all the code snippets and all the teaching material that you will need to effectively follow this course. Now, this repo is under constant change, so you will find content changing every now and then, depending on the challenges or requests I receive in this repo. Now, I want to highlight a few important things when working with this repository. All notes that will be necessary for you are included in the advanced.sh file. This file contains all the notes that explain the various pieces of codes that we will be using for this course. For example, you will find that I annotate what we're going to do or the steps we're going to follow, write the code down, and then in some areas, I quickly explain what the code does. For example, here I explain what the loop does in this code snippet. Okay, I also go further to try and explain each and every component of the code that we'll be using. For example, I explain the need for including -r here and just other parts of the code that you will be using. Now, when you head over back to the repo and access the file called main.sh file, you will pretty much have all the steps aggregated together, but without any annotation. This is simply an end-to-end pipeline that you can easily run in your computer and pretty much achieve the results that we will achieve at the end of the tutorials. Now, take note that for the bash script tutorials, we'll mostly be using the .sh files. And that is why you will find the main.sh file as well as the advanced.sh file in the repo. Now, this applies the same when we will be learning about the working with Snakemake and using Snakemake utilities. For example, you will find that I have included the main.smk file that contains an end-to-end pipeline using Snakemake. So you can easily run this file in your computer and pretty much achieve the same results as if you ran the main.sh file. I also include a file here called prim.smk, which pretty much contains all the annotations that you will need and some notes and guidance on what you will be doing. For example, I explain why we use the rule as a Snakemake utility in this file. I also include some housekeeping notes just to get you started and going with the tutorial. Now, one more thing I would like to highlight is that I included the srr sec list.txt file, which we will use to store all the accession numbers for the samples or the raw FASTQ files that we will be using. Within this repo, I also include the tool installation.sh just to keep all the installation scripts for all the tools that we'll be working with in the entire lecture. Now, that's it for the repo. You can access it there. If you have any problem with the repo, you can easily create an issue and I will quickly go through it and provide a response just in case. Now, the other way to access the resources that we'll be using throughout the course is to head over to my Medium blog. I have included a link in the video description and you can simply click on that link and just read the tutorial there. Now, the blog is quite different from the notes I've included in the GitHub repo, and I feel like there is much more clarity and you don't need to kind of struggle with navigating the hash outs or any other things that pose as a limitation to using the GitHub. Now, with that said, let me head over to my Visual Studio Code to show you just a flow of how we'll be working throughout the entire series. During my tutorials, I'll start by giving the simplest way of doing the analysis and then proceed to show the best and the most advanced way of doing the analysis in Bash. And just to show you throughout the lecture, what we'll be aiming to do is to build this workflow from top here to the bottom. This will give us a good foundation on Bash that we can now use in Snakemake. Now, that's pretty much it, and I hope you will enjoy the course. And if you are liking these lectures and liking this series of tutorials, I will request that you give me a subscribe and you give me a like in the video and share the video with your friends or in your platforms. And you can also recommend to me any future videos that you may want to learn or any concepts that you may wish to learn. If you have any projects that you would like me to collaborate on, I also welcome you to contact me through my emails. Otherwise, thank you and see you in the next video.